Hey everyone, my name is Hannah Leopard. I am with the Growing Genetics Center, specifically the Education Department. And this month we are going to be virtually celebrating DNA Day all month long. And for this week, we're going to be focusing on the fundamentals of heredity. So for starters, we need to talk about what heredity is and what that means. So obviously the first person that we're going to talk about is Gregor Mendel. So he is essentially known as the father of genetics, and that is because in the 19th century, he actually was the first to discover the principles of heredity and how traits were passed down by using pea plants. So the easiest way that we can track how traits are passed down is to use what's called a Punnett Square. So we are going to talk a little bit about that today, and we are also going to talk about some of the terminology that's involved when using Punnett Squares. So for starters, when we talk about alleles, alleles can either be dominant or recessive. So we essentially have two alleles for each trait. We get one from mom, one from dad. So when we talk about alleles, we actually are going to use uppercase and lowercase letters to represent these alleles. So for a dominant allele, we're going to use an uppercase letter, and I'm going to use the letter A. For a recessive allele, we're going to use a lowercase letter. So I'm going to do a lowercase a. So for our Punnett Square today, we're going to practice doing a cross by doing Gregor Mendel's cross that he used between his pea plants. So he crossed a yellow purebred pea plant with a purebred green pea plant. So green was the recessive trait for these pea plants, and yellow was the dominant trait for these pea plants. So if we cross a yellow purebred, we're going to do a capital A and a capital A. And with a green purebred, we're going to use two lowercase a's. All right? So if we cross these two plants, we can actually look at the probability of what the offspring may look like. So you get an uppercase and a lowercase letter here, an uppercase letter here, and a lowercase letter here, uppercase letter, and a lowercase letter, uppercase letter, and a lowercase letter. So if we wanted to write out the probability of all of these plants being yellow, we would confidently say that 100% of the progeny would be uppercase A, lowercase a, and yellow. All right, so let's say we wanted to find out what the next generation would be, what the probability of those plants would look like. So what we'll do is we'll take our generation above and we will cross them. So we'll do, remember, an uppercase A and a lowercase a, and then we'll cross that with another uppercase A and a lowercase a. So if we want to write out and find the probability for this generation, what we'll have is an uppercase A, uppercase A, uppercase A, lowercase, uppercase a, lowercase a, and a lowercase a, and a lowercase a. So this one definitely looks different from our first generation. So if we want to write out the probability here, it's going to be a little different. So for our first box, 25% were capital A, capital A. Our next two boxes, which would be 50%, were uppercase A, lowercase a. And then our last box, so the remaining 25%, were lowercase a, lowercase. So if we want to write out the phenotype for these individuals, remember that in uppercase A means that we're going to see yellow pea plants. For an uppercase A and a lowercase a, the uppercase, remember this is a dominant trait, we are also going to see color yellow. Whereas for the remaining 25% of the individuals produced, we had a lowercase a and a lowercase a, we are going to see green pea plants. If we want to go one step further, we can actually write out which individuals are heterozygous and homozygous. So those are the last two terms I want to talk about today. So we use the term homozygous to represent an individual that has the same two alleles. So they're going to either be capital A, or lowercase a, lowercase a. So you can either be homozygous dominant or homozygous recessive, depending on your two alleles. Whereas you could also be heterozygous. So a heterozygous individual will have one uppercase A and one lowercase a. So if we were to designate these two terms to our individuals, what we'll do is we will say that these individuals were homozygous dominant these individuals are heterozygous because they had two different alleles, and these individuals would be homozygous as well, but they would be recessive because they contain both recessive alleles. So when talking about heredity, Punnett squares are a simple tool that we can use to determine the probability of certain traits being passed down to future generations. Thank you guys for tuning in and watching Heredity with Hannah. Be sure to like our Facebook page and be sure to follow along as we celebrate DNA Day virtually all month long.